Good morning, all of you. So today we are going to learn about the basic concepts of plant tissue culture technique. Let me share the slide and then start. Hope you all can see my slide. So plant tissue culture, we are going to learn about the basic techniques of plant tissue culture. Let me see the definition of plant tissue culture. So plant tissue culture is a technique of growing plant cell, individual cell or group of any kind of tissues or complete organs or seed, non-viable seed or any other plant parts other than what I mentioned. So this we call it as plant tissue culture technique. Plant tissue culture technique mainly depends on two major things. One is totipotency. The actively dividing plant cells, what we call it as meristematic cell, have the capacity totipotency. So each individual cell of highly meristematic nature can able to regenerate into your whole plant if you are providing a proper nutrient in aseptic condition, especially in plant tissue culture. Second one is plasticity. So you all know plants are autotrophic mode of nutrition. It can able to synthesize their own food by the pigments present in the chloroplast. When you are taking a piece of tissue to regenerate any plantlets under plant tissue culture laboratory, so that time those plants are partially heterotrophic because you are giving a new environment, not exactly the soil. So that time the plant tissues have the capacity, ability to plasticity, can able to alter the metabolic pathway, growth and development differentiation of those tissue to best suit their environment which is given in the plant tissue culture. So plant tissue culture mainly depend on the meristematic cell nature of totipotency and plasticity. The ability can able to alter the mechanism, metabolism, growth and development what you have given in the plant tissue culture laboratory. So moving on to why plant tissue culture we are using. So there are varieties of application and reasons why we are using plant tissue culture to clone more number of plants in a short time. If you leave it by nature in the soil, you can't able to get much number of plants within a short duration of time. It will take time because abiotic factors, some environmental factors also having some effects on the plant. So that alter the maturation time. So by using plant tissue culture, you can able to produce more number of plants within a short duration of time. And second one, to enable the plantlets to produce desirable traits. Desirable traits means plants having the compounds, the economic value compound. So that compounds can be produced in a good quality, desirable price. Say for example, if I'm generating tomato plantlets by using plant tissue culture. So if I'm concentrating the lycopene content of the tomato, so from my plant tissue culture tomato plant, if I can able to help the society to get more lycopene content, that is the desirable trait. Okay, so like that, whatever vegetable crops or anything, economic value, economical crops, whatever you are using in plant tissue culture, if you can able to produce a desirable price to the economic value to the society, so that also is very much possible by plant tissue culture technique. Third one, to quickly produce mature plants. Okay, so within a short lifespan, you can able to produce fruits, flower if you are raising the plants by plant tissue culture technique. And fourth one, to multiply plants in the absence of seeds. Some plants like orchids, nepenthes, not only that, rare and endangered plant species, if it can able to produce seeds in large number, if those seeds are non-viable, so those plants, any of the plant tissues you can able to choose and you can able to raise the plantlets by plant tissue culture technique. The next one, possible to produce plants from seeds having very low chances of germination. If the seeds are having very low chances of germination to start the second generation, those plantlets you can choose. Instead of going for the chance of seeds, you can use any of the plant tissues to raise the plantlets by using plant tissue culture technique. The last one, disease-free plantlets can be quickly multiplied from disease-spot plant. If I am choosing any rose plant or any apple plant which is the stock from my garden, if it is diseased one, if I have chosen any disease free stock piece of stem or root or leaf, so from that I can able to produce disease free healthy plant in my plant tissue culture though my stock plants are diseased. Okay, so these are the reasons why we are adopting plant tissue culture technique. Okay, so this is the basic mechanism how the regeneration, metabolism modification happens in the plant tissue culture bottle, the plant tissue culture technique. Here it is shown the carrot tissues. These are the highly meristematic cells. We have to isolate in an aseptic condition, complete absence of microbes. That's why it is aseptic, absence of septic by microorganism. So if you are using the highly meristematic divided cells, you have to inoculate in the tissue culture bottle or slant, okay, and keep it for incubation. After that, I told you each individual meristematic cell have the capacity, totipotency. So because of that, the proliferation, continuous division happen and it will produce a disease-free healthy plant nest. Okay, so this is how the because of the pluripotency nature of the highly dividing meristematic cells, we are running still successfully the plant tissue culture technique. Not only for carrot, so all the plants having meristematic cells in the meristem shoot tip or in the node region, end stem, end leaf, end root. So all the samples you can able to raise the plant test. This is an example I have given you as a carrot tissues. Okay, then plant tissue culture can be divided into two types. One is organized plant tissue culture, other one is unorganized plant tissue culture. See on my left hand side, you can able to see the complete organs of the plant. Buds in the node, meristem, shoot tip, embryos. Embryo no need of culturing in plant tissue culture in case if the endosperm, the white part, so for example coconut, the white part is the endosperm. If the endosperm is not well developed properly, that is a beauty tissue giving nutrition to the developing embryo. If the endosperm is not well developed properly, the embryo will be suffering. So that time to rescue the embryo, you can use those embryo, we call it as embryo rescue technique. Then non-viable seeds, anther, pollen, ovary or ovule. If I'm using complete plant parts, that is coming under organized plant tissue culture. So on the next side, unorganized mass. Unorganized mass means any clump of simple parenchymatous cells. If I'm using unorganized means I cannot able to predict which portion is going to be the shoot, which portion is going to the root. Okay, so that's why the name is given unorganized mass. Here the callus. This is unorganized mass of simple parenchymatous cell. Today, if I am keeping one piece of stem from rose plant. After my incubation time, what happened? The cut edges of the rose plant come in contact with the media, so that produces a tumor kind of cell. It is not always possible to give the tumor kind of cell. It depends on the genetic makeup of the plant and the composition of auxin and cytokine, those both growth regulators added in the medium. So this is the callus. If I am using only one callus, I may get only one plant. If I chop the callus into many pieces, see the next slide. Next picture, that is cell suspension culture. How many pieces I chop? Each pieces, if I do the subculture, I can get many plants. And third one is protoplasm. Without cell wall also, with the protoplasmic content, you can able to rise the plant. This is also one choice of plant tissue culture. So without cell wall also, the meristematic nature of the cell, the totipotency, it won't affect. So protoplast also we can able to raise the plantlets and somatic embryo. Embryo-like cells. You might have studied about various stages of embryo like heart shape, torpedo shape, quaternary embryo, then spherical shape. So many shapes are embryo you might have studied. So from the callus, the white part is the callus. From the callus, it raises somatic embryo-like structures. So those embryo also you can excise from the callus and you can do the subculturing. And here the direct organogenesis without going for callus, if the explant directly produces organ, so that also coming under unorganized plant tissue culture. So plant tissue culture can be divided into two types. One is organized, another one is unorganized. Okay, then moving on to the explant. Explants are nothing but excised plant parts. If you're taking the pieces of leaves, then cell devoid of cell wall, then a piece of nodal segment, then piece of internodal segment or piece of root, pollen, anther, ovary, ovule. So those pieces we call it as explant. Excised plant parts we call it as explant. This is how the regeneration happens when you are using these many types of explants. Okay. Then moving on to steps in plant tissue culture. So the major steps are these are the you have to choose the piece of plant from your disease-free healthy stock plant. Then you have to surface sterilize it. You have to wash it properly. There are some detergents to wash the explant surfaces. Okay, then you have to inoculate it in the plant tissue culture water containing media. You have to incubate it. Okay, so after a certain period of incubation, in vitro plant, some differentiation happens because of the totipotency nature of the enzymatic cell, which is having it in the excised exercise the plant part which you kept it in the tissue culture bottle, and after that you should not wait till the plantlet reaches the neck of the bottle or until the nutrient exhausted. That will damage the plantlets. What happens before the neck of the plant bottle, you have to take it out and you have to put it in the pro place. Have a cocoa pit and keep it under your observation in plant tissue culture laboratory for 15 to 20 days. So still that if the plant is still healthy, then after 20 or 15 days you have to plug the cocoa pit along with the plantlets and you have to go for soil. Okay, so this is the hardening or acclimatization technique. So these are the basic techniques since we are running the plant tissue culture successfully. Explant chosen, surface sterilization of the explant, inoculating, subculturing, in vitro differentiation definitely happens, then primary and secondary hardening under your supervision for 15 to 20 days, and finally you have to transfer that plant tissue culture disease-free healthy plantlets to soil, or you can say greenhouse condition. Okay, then factors affecting plant tissue culture. So what are the factors that will affect your result of plant tissue culture? First one is genetic makeup of the plant. So how the genes are responsible to alter the result from your explant. Then age factor. Whenever you are choosing any explant, I told you it should be disease free, very young meristematic cell you have to take. You should not take the adult or old one. So age also one deciding factor for the success of the plant tissue culture. Third one is type and source of explant. What type you are taking. Okay, whether it is a parenchyma thistle or from shrub you are taking, herb you are taking, that decide, and source of the explant. Source means which portion you are taking, leaf or protoplast, anther, pollen, ovule, embryo, root portion, internodal portion, nodal portion. So that also will determine the success of the plant tissue culture. And fourth one is nutrient media. There are specific media to raise the plant tissue culture based on the extent which nutrient media you have selected. Okay, so these are the four factors that determines the success of your plant tissue culture. Okay, then application of plant tissue culture. We have varieties of plant tissue culture. I will name a few. So yearly flowering can be achieved quickly. The plant will mature. Then fast multiplication, genetic transformation, symptomatic embryo differentiation, large scale multiplication of plants, secondary metabolite production from the plants. Then disease-free plant, you can able to alter the number of chromosomes. So many varieties of application this plant tissue culture technique have. Okay, so with this I stop my lecture. So I hope from this lecture you get some concept about some idea about plant tissue culture technique. See you all. Thank you.